Welcome again to this guide to the maintenance of life science data by biocurators. My name is Claire Donovan and I have been in the biocuration field since 1993. Next slide, please. So the aim of this webinar is to introduce you to the principles of how biocurators archive, curate, maintain, and update life science data. And if you're from our user community, how we can work together to ensure the data is represented in the best possible way. Next slide, please. So the format of this is 4 talks. Firstly, I will give an introduction and overall view of a biocurator role, which varies between resources, but has general principles in common. And then I will hand over to 3 of my lovely colleagues at the EBI who work on different resources, who are going to focus on certain aspects of that role to give you deeper insights into what we do every day. So Birgit, Deborah, and Rosanna will follow on. And next slide, please. So what do biocurators do all day? Next slide, please. But firstly, we have to discuss who is a biocurator. And the most important thing you need to realize that she or he is a scientist who extracts, interprets, combines, and archives the data. It's critically important that you understand that the person at the other side of that resource is interpreting your data, interpreting the data from the publications from a whole variety of sources, and then combining it all together and making it machine readable, standardizing it, making it machine readable and more discoverable and accessible to the public. So it's a very important job. So what makes a good biocurator? You need to be more than anything else detail-oriented and conscientious. People use these resources, they depend on them, and we need to do the best possible job that we can. You also need to be a very good communicator with diverse user communities from all different fields of science and from all over the world. And usually you will also be embedded in a team with software engineers and bioinformaticians who will not have any domain expertise, but you need to work together to make sure that you explain the data to them, they explain to you what the technologies are, and together, working as a team, you make the best resource possible. And I find this dynamic working environment incredibly rewarding. Usually a biocurator has a PhD in their subject matter of expertise, but another critical thing to convey to all of you is that no one ever knows it all in the field of biocuration. It's constantly evolving, so you need to be open and flexible to learning new things because both the data and the users continuously evolve. Next slide, please. So now we'll go into some of the typical job requirements. So I don't want to use the expression piggy in the middle, but quite often that is what you are. You deal with all the stakeholders of the data, from the producers and submitters of the data to the consumers of the data on the other side. And you need to make sure that they're all happy and that the data is both provided and utilized in the best possible way. You also need to have an understanding of the wide variety of data types. You might not deal with all of them, but your data will be interacting with them. And it's important that you understand the ecosystem in which your resource is sitting. There are other issues about the nuances regarding the handling and storage of this type of data. Depending on how the data is being exposed, it needs to be stored in a certain way. So you'll be working with that software engineer and bioinformatician to make sure that it can be queried in the best possible way, disseminated in the best possible way. And this can be very exciting to look into. Another aspect of the data, when I say we combine it and we curate it, is that we take the data that the submitter uses it and we add metadata on top of it, such as ontologies, that make it interoperable with the rest of the resource. And you'll hear more detail about that later in the webinar. Next slide, please. So again, understanding the latest technologies for the storage of biological data. We're all aware of websites changing all the time, but so do databases, and how you can best store the data and scale the amount of data that's available now compared to where it was only a few years ago is incredible. And you need to make sure that your data is being stored in the best possible way for it to be actually utilized, both by bioinformaticians and by scientists who just like to sit there and read it. Because this is constantly changing, you need to be able to self-train, to sit there and learn. There are no courses really. You just have to sit and be proactive and work with your colleagues to make sure that the data continuously is provided in for the maximum benefit of our users. Another critical role is an understanding of the life cycle of the data. In particular, the storage of the data for later reuse. So you take your study or your record, you work with the submitter to make it the best possible way. You provide all the evidence for why you came to the conclusion of whatever it is that you're presenting in that particular record. And then you put a timestamp on it to make sure that when someone comes to use the data, they understand when it was done, what possibly came after, and the levels of evidence that were attached to it. Again, you will get this in more detail later. Next slide, please. So more recent challenging parts for curators is understanding the various data license types. Traditionally, academic resources such as those at the EBI are free to use by anyone and everyone, but it is increasingly common now for biocurators to work in more commercial environments or semi-commercial environments, so that you need to be aware of the licenses, either if as a user or working with those biocurators, or as someone who actually works in a resource like that yourself. Another very challenging thing which has happened in the last few years is understanding how to best manipulate human data. So there are privacy laws that are very specific to separate countries, continents, even states within a country. And it is important for you always, if you're working for such a resource, to be aware of your responsibilities, but also if, as we are working for an archive, if someone submits data to you from a country that you know shouldn't be submitting outside their own country, it is important that you're aware of those and reach out and not accept the data, basically, and make sure that that's clear to everyone involved. So these are the more interesting aspects of how the biocurator role has evolved in the last few years. Next slide, please. So in summary, biocuration involves the translation and integration of information relevant to the life sciences. Lots and lots of information, diverse information, into a resource that can then be used by those diverse communities for multiple purposes. I have worked on a number of resources over the years, and I am constantly surprised by what they're actually used for. You think you have one user base, but you have very many. And what we have also learned is that we provide such essential resources to the life science community that resources have become an integral part of the tools researchers use on a daily basis for their work. So it's really, really important that we as biocurators do it in the best possible way, and that we work with our communities, who I hope some of you here represent, to make sure your data is represented as accurately as possible and most completely as possible. So that's the overview, a whistle-stop tour, but the slides will be available afterwards. And then I will hand over to the first of my colleagues, Birgit, to go into the details.
Yeah, hello, my name is Birgit. Thank you for the great introduction, Claire. I'm a curator at the EBI, and I work on protein interactions and complexes, but today I will focus on the importance of data standards and how you can help us to keep all of this data, well, nicely standardized. Next, please. So data standards are everywhere, from globally recognizable ISO units like the kilogram or the meter to domain-specific formats like domain-specific standards like the FASTA format for molecular sequences here on the right. Next, please. And without standards, there's no fair data. Many of you will have heard about fair data, and Deborah and Rosanna will go further into it. And in order to make data fair, we have to have standards. Next, please. Okay, standards come in two flavors. There's standards for entities. So they provide unique identifiers for things like proteins or genes, or PubMed identifiers for publications, or even domain-specific dataset identifiers. And secondly, standards describe terms that we use, terms that describe the entity again, or terms like describing molecular functions of a protein, or the experimental parameters that are necessary for an experiment. And essentially all metadata can be described in terms of standard terms. And in order to organize these, we use control vocabularies and ontologies. Next, please. Control vocabularies define the terms. A term is just a name, and these names can evolve. And therefore we have synonyms as well. So they can include alternative spellings and historic terms. But the definition is what makes a term standardized. And they all get unique identifiers. Next, please. So in this example, this is a cyclin-dependent protein serine threonine kinase activity definition. The term has got a unique identity given by the gene ontology, which is the domain authority for molecular functions. And the definition describes these functions in terms of its biochemical reaction it catalyzes. Next, please. And because this is a very common name, it's a function that's been around for a long time. It has accumulated a lot of different types of synonyms. And it's even got an alternative ID from when two terms were merged. And all of these IDs and synonyms, when you use them to search, will come back to this one standard entry. Next, please. When we combine all of these CVs in one subject domain, we can build ontologies, which are the hierarchical representation of these CV terms. So in the case of the cyclin-dependent protein serine threonine kinase activity, it puts it into its biological function context and describes it as a protein kinase activity, which is also a phosphotransferase activity, or a kinase activity, which is a transferase activity, which is a catalytic activity. So this hierarchical structure for terms allows us to do two things. A, we can annotate any molecular entity, such as proteins or genes, with the most specific or granular term that's available or that we experimentally know of. But we can also search data by using any level in the ontology to find all protein kinases or all cyclin-dependent protein serine threonine kinases. Next, please. So I'm going to give you a little glimpse into how we as biocurators extract these standard terms from a piece of published information. So a paper that contains text, figures, tables, appendices, supplementary data. Next, please. So in this case, I'm trying to extract the molecular interactions that are described in this publication. And at first glance, it looks as if the authors are making our life easy by mentioning the two proteins, CDK9 and cyclin T1, already in the title and the abstract. But at second glance, that's not enough. They don't mention the species, and we know CDK1 and cyclin T1 are found in many different species. And it doesn't describe them as a unique identifier. Next, please. The species we can identify from the publication. If it's not in the publication, we have to go through references, etc., which can be quite cumbersome. But here we're lucky it's mentioned once, so we're dealing with human CDK9 cyclin T1 complex. Next, please. And then we have to find the actual standard identifier for these entities of two proteins. And for that, we go to the authoritative database for proteins, which is UniProt. And because UniProt captures all the different aliases, names, synonyms for different proteins and the genes that code for these proteins, we can use these symbols as a search term. Apologize, it's a bit small, but I searched for CDK9 and with SUCT1, which were the two symbols mentioned in the publication. And then I get search results for each individual species where this protein has been characterized. In our case, human comes up as the first, the top hit. And the standard identifiers in the first column. The other thing to notice here is that for cyclin T1, the symbol or apparent symbol that was used in the publication is not even the standard gene name. Turns out the standard gene name is CCNT1, which wasn't even mentioned in the paper. So, but because biocurators manually provide this information when they create the entry, we can then find it when we need to use it in other curation. And we can transfer all of these identifiers from one database to another to link it all up. Next, please. So that was an example of how we find an identifier identity. And now I will show you one example of how we find information about metadata. So for molecular interactions, we have to put them into the context of what experiment they were conducted in. And this paper, at least one of the experiments, was done by crystallization, which was quite straightforward to find from the text. But crystallization is just a name. So again, we need to find the identifier. Next, please. And for that, we use the ontology lookup service, which is publicly available and contains nearly 300 biological ontologies. And it's making our life very easy because you can just write the term that you're looking for into the search box, and then it suggests different terms and in different ontologies. So for the trained biocurator, we know which ontology is applicable for our domain. In my case, the molecular interaction ontology, here the MI in the middle of the suggested hits. And then I can take that term, I can take the UniProt identifiers, and all the other terms that I pull out from the controlled vocabularies and ontologies, put them into our editorial tool and create the entry. Now we do this for all the entries, and at regular intervals, like Claire already mentioned, this data gets published. And then you can make use of the hierarchical structure and the use of these standards for finding the data yourself. So next, please. If we go to the INTACT database, which is the molecular interaction database at the EBI, and we search with the UniProt identifier for CDK9, which is P50750, we find 552 interactions. As you can see from this overview, these interactions are not just with proteins, but also other molecules, and not just with human proteins. We were interested in the evidence from the crystallization experiment, so we can even use the search function in the network graph to pull out its interactor, CCNT1, or using the UniProt identifier. And then we can pull it out, which I've already done here in the screenshot, and you can see that 26 different evidences for this interaction. The table below shows you a lot more information about each interaction. The second line is the evidence for this crystal that we looked at. The first line is a completely different evidence for the same interaction. Only because we use the standard UniProt identifiers do we get the whole list of these interactions in one view. Furthermore, you can see that most of these terms are hyperlinked. If this is a standard entity like a UniProt ID or publication, these link back to the primary databases that capture that type of data, so UniProt or EuroPubMed Centre, for example. Other terms then link back to the ontology lookup service, where you can check the definition of these terms if you're not familiar with them. So for example, the first experiment that's in the table was conducted by Antibate Co-IP. So if you're not familiar with that particular experimental method, you just click on the link and it gives you the definition. You can also use all of these terms to filter the data further and provide a customized result based on your particular research needs. Next, please. So overall, what are our challenges when we want to standardize our data? So it's identifying the unique identifiers for things like proteins that are just identified by a name or symbol in the text, identifying the species. Sometimes we can find them by references or by contacting the authors, but sometimes it's just impossible to identify the species from the publication, and then that data cannot be curated. We also often deal with data in PDF format. For example, large-scale data sets are supplied in the supplementary data as PDFs, and we end up having to manually copy-paste the data out of it. And of course, data knowledge increases, knowledge emerges, new experimental methods are developed. So CVs and ontologies are living systems. We constantly update them to fit the data that is actually available and the data we need to capture. So I have essentially two pleas. One, please describe your experiments in much detail as you can, and use functions like extended methods in supplementary data. And also, even if your data doesn't have to be submitted, like sequences or structures, you can still submit your data to databases directly, which allows you to have a conversation with the curators so we can capture the data correctly and in its entirety. So that's all from me, and I'm handing over to Deborah, who will explain a lot more about this communication between curators and submitters. Thank you for your attention.
Thank you, Birgit. So my name is Deborah Hers. I work as a biocurator for the Protein Data Bank in Europe, and my part will focus on the user interactions for the benefit of both the data and the submitter. Next slide, please. So I'll begin with a short word about the Protein Data Bank Archive, or shortened PDB. It's a single repository of 3D structures of proteins, nucleic acids, and complex assemblies. And it's a rather old database since we celebrated its 50 years anniversary this year. So obviously, the database had to evolve and adapt a lot in terms of file formats and data content as the technique evolved as well. So the archive currently has over 170,000 entries covering more than 60,000 unique sequences and over 30,000 unique ligands. Next, please. So the PDB is a worldwide archive with people working from 3 different sites, one in the USA, one in Europe, and one in Japan. And we are 20 biocurators in total, working from those 3 sites, which requires that we coordinate regularly to make sure that we share the same biocuration practices and that depositors from anywhere in the world will receive the same service and the same annotation of their data. So last year, we annotated over 17,000 entries and created 3,000 new ligand definitions. Next, please. So what kind of data do we curate? Mainly 3 things. The first curation consists in mapping the chemical components, so ligands, to the chemical component dictionary. Or if it's a new ligand that comes in, then we create a new definition. The second curation concerns sequences, where we map protein sequences to the UniProt database. As it was mentioned before, this is a reference database for which we use the identifiers. And nucleotide sequences are mapped to GenBank. And at this step, we also enrich the sequence annotation by adding any information regarding expression tags, engineer mutations, deletions or insertions, etc. Next, please. The third main added annotation in PDB entries concerns the assembly information. And this one is still mostly a manual process. So for this part, we cross-check the information given by the depositors and we use our own expertise to do the annotation. So this annotation, the assembly one, as for the sequence and the ligand annotation, is reviewed by the depositors after annotation. So what happens is that after the annotation process is complete, we send a validation report to the depositors, which allow them to control the quality and the accuracy of the annotation that has been done. And what is interesting is that the very same report is later on available to any user of the database so that they can assess both the quality of the data and the full content of the curation. Next, please. So one interesting thing about our archive is that the life cycle of an entry can begin even before the scientists deposit their data in our database. So indeed, we provide a standalone validation server as well as a validation API so that the users can validate their data before making the deposition. So how it works is that the scientific community defines this data standard that we use for validation, and we update our validation server regularly and accordingly to a scientific community opinion. Therefore, the depositors can always validate their data against the latest data standards. Next, please. Another interesting aspect of our database is that we allow round trips of deposition and curation. So not only we focus on harvesting as much metadata as possible during deposition, but also after curation, the depositors are asked to review the accuracy of the curated information. And if they have corrections to make, we allow several round trips until both the depositors and the curators are happy with the outcome. So this process is beneficial for depositors, of course, for the quality of the archive, and therefore for the end users. Next, please. So I would also like to mention that even after the data is released in our database, the users can still come back and update their entries. So for example, they might have produced a better model to explain their experimental data, and they want to replace their old model with the new one. And this is allowed as long as the experimental data isn't changed. Another example would be the update of the citation information. So once a publication is out, all the users can come back and say, can you please update the citation information so that the two are linked. But they can also update any other metadata. So for example, oh, I just noticed I've introduced a typo in my colleague's name, or, oh, actually the mapping to Unicode was incorrect. Can you please fix it? And we can do all of that afterwards. And as I mentioned that the data standards can evolve over time, we can carry out remediation projects to update parts or whole of the archive. And this is what we did recently when we changed the way carbohydrates are stored and represented in our database. And this stands true also for the validation reports, which are recalculated on an annual basis so that they always display the most up-to-date validation values. Next slide, please. So my final slide is to mention that curation is a very rewarding job. Biocurators are expert scientists who provide a service with lots of human interactions, and those user-curator interactions benefit the data in order to make it fair, so findable, accessible, interoperable, and reusable. So I will now hand over to the last speaker, Rosanna.
Thank you, Deborah. So I'm Rosanna. I'm one of the curators at Uniprot, And today I'm going to give you an overview on how we curate literature in UniProt. Next slide, please. So literature curation in databases play an essential part for many scientific resources. It is the principal source for experimental evidence for the functional characterization of a wide range of biological entities. And here you have some of the databases and which type of biological entities they curate. And also it's playing an important role for machine learning and text mining. Next, please. So to illustrate how literature curation works, I will use the example on how we do that in the UniProt database. So UniProt is a comprehensive, high quality and freely accessible resource of protein sequence and functional information. We have more than 200 million protein entries that cover a large variety of species. At the last release, we cover more than 800,000 taxa. And literature curation play an essential role in UniProt. And indeed, we have more than 400,000 manually expert curated papers, and we also add more than 1 million computationally mapped paper. Next, please. So on the left, you have an example of the typical workflow that we use to create an entry in UniProt. So the first step involved the selection of an entry from the unreviewed pool in the database. So we have various criteria to select an entry. We prioritize the curation of proteins from human and model organisms. We also curate proteins where the user requested an update or the addition of a publication, or because they found an error, which can happen. We also curate protein that comes from our funding. For example, this year we have curated several proteins that are linked to the Alzheimer and Parkinson diseases. We also scan the literature weekly to find protein where a new function has been described. And also we curate proteins that are important to current health issues like the current COVID pandemic. We also go to conferences to see what are the hot protein at the moment, and also we curate protein based on our own expertise. For example, I specialize in the curation of enzymes. So once we have selected an entry, it goes through what we call the expert curation process. This involves the literature curation, which I'm going to talk about in more detail in a moment, the sequence curation, the annotation, and then once this is done, the entry is integrated into the reviewed part of our database. But these reviewed entries are not static. It means that they are continuously updated as more knowledge becomes available. Next slide, please. So what type of information do we extract from the literature? For a UniProt entry, quite a lot. We extract protein engine names, the function of the protein, obviously, its catalytic activity for enzymes, the subcellular location, its expression, with which protein it interacts. And we also curate information for the sequence of the protein, like post-translational modification. And for human protein entries, we also extract information about diseases and variants. Next slide, please. So this is a typical UniProt protein entry. And here you can find all the various topics that I just mentioned. So at the top, you have the unique protein identifier that Birgit has mentioned earlier. And here I have highlighted how we add the publication to each piece of information that we provide in the database. We have also a a dedicated page for publication that you can access from the top left menu. Next slide, please. And here you can find all the paper that have been curated manually, and they are classified into categories. And we also provide computationally mapped references. Next slide, please. So what curation of literature involves? There are quite many challenges. First, you have to find and select relevant papers. Then you have to critically evaluate the literature. Then you have to extract and compile all the relevant information, and if necessary, reconcile any conflicting results. And at the end, you have to provide a link to the original publication. Next slide, please. So how do we find and select relevant paper? This is quite a time-consuming step. So we use keyword-based query method. So we use gene name, protein names together with the species to query literature database such as Europe PMC or PubMed. And here, once we have all the potential paper, we decided which one to exclude. For example, we exclude reviews. Although I have to say that we read them as they provide background on the protein function, and sometimes they provide additional reference that we could have missed with our search, but we don't add them to an entry. Also, if a paper has no protein related information, for instance, if they have characterized the promoter of the gene, we don't consider this paper. And also, usually we don't annotate high throughput papers. So for each paper, we read the abstract, we scan the full text, and then we select the most relevant publication to avoid redundancy, and then we read them fully. And to give you just an idea of the scale of the work, so about more than 8,000 papers are curated each year. This represents about 200, 300 papers per curator, but each all the curator will evaluate more than 50,000 papers per year, which means that one curator will go through about 2,000–3,000 papers a year. Next slide, please. The second step involves the critical evaluation of all the papers that you have selected. Here you will assess the strengths and the quality of the evidence, and this is based on the curator lab experience. And this is invaluable to do this. Also, we check if the data is supported by available experimental evidence. Sometimes we have statements in paper that are not supported by a picture. They are the data not shown, and it will be really nice if user can put this data not shown as figure in supplementary materials, because otherwise we won't curate them. Also, we check if the controls are appropriate for the experiment, if the biological system used is adequate. Next slide, please. Then we extract all this information and we compile it. We try to be as much concise as we can. The idea is not to write a review. And also we try to reconcile any conflicting results. We also try to standardize the capture of knowledge, like Birgit has shown you before, to make it more searchable afterwards. And you can use various ways to annotate your entry, and this will depend on the database. But in UniProt, we use free text, and here you have an example for the function comment. We use also control vocabularies, such as for the subcellular location section, and ontologies like the Go annotation, the gene ontology annotation system that Birgit mentioned earlier. Next slide, please. And the last step of the annotation process is to provide for each piece of information that we add the link to the original publication. So as you can see, there are many challenges in the curation of literature. And as I said at the beginning, one of the main challenge is to find relevant publication. And for this, the community can help. Next slide, please. Especially by structuring knowledge when they publish their data. For example, by using identifiers like official gene name, protein ID. And here you have a very nice example of a recent publication where the authors provide two unique identifier for the protein, the UniProt ID, and also an identifier from the database specific for the species. Also, several proteins are spliced, so it is important to specify which isoform has been used for the experiment. And also, this is very important to specify in which species the work has been done. And for bacteria, viruses, parasites, and fungi, it is also important to mention the strain. And in the end, all this will facilitate text mining. And for us, we will find much more quickly the relevant publication for the protein that we are curating. Next slide, please. So another way that the user community can help us curating entries in UniProt is by adding the publication via our bibliography submission page. And here, user can add a paper. It doesn't need to be their own publication. It can be any type of publication that they find interesting, and so they can add the paper, but also provide some basic annotation extracted from this paper. Next slide, please. And this will then be available in the publication page for the protein entry under the community curation. Next slide, please. So I show you how we do the literature curation in UniProt. I hope it was useful. And we hope also that these three examples that Birgit, Deborah, and I has provided you with a better insight of some of the aspects of the work of biocurators, and especially how essential this work has become with the avalanche of biological data that is happening since more than 30 years. And with all that, I would like to thank you for listening, and I hand over to AJ.
Thank you very much, Rosanna. And thanks to all the speakers of the day, Claire, Birgit, Deborah, and Rosanna. So before we go on to Q&A, I would just like to say that this webinar was part of our A Guide to Webinar series, which we have been running since April. So if you're interested in what topics we cover, so you can visit our training website. Next webinar in this series is about proteomics data analysis from UniProt and Interpro, which you can look, you might find it interesting. And if you want to contact any of the speakers of the day, you can write to them directly, their email. And we'll share this slide as well with you along with the recording. So you can get the email IDs again from the slides. So with that, I will now hand over to Claire to facilitate the question and answer. So you can keep writing your questions in Q&A box.
Okay, so one was from Andrew Bandral, who said, how is it to work as a biocurator? And I said, well, I love it. I'm still doing it. But I would also like to open that up to Deborah, who's one of our newest curators first.
Yes, sure. Well, it's just awesome. Every day is different, and you get to talk with a lot of people and see a lot of different data. So if you're interested in, you're interested in your field, obviously. So you get to see the latest advanced in your own technique, which is really superb. And on top of that, you get to communicate with the depositors, which is a real pleasure. So yeah, totally recommended.
Okay, and then to Birgit, who has one of the longest with us.
Not as long as you, Claire.
Yeah.
Yeah, it's similar to Deborah. It's knowing that you can make a difference with every entry that you push out. So it's a bit of a faster turnaround time than in the lab, for example. There's always something to do. And also it's so varied. So in my field, I work with a lot of other teams. So I work with the PDB team that Deborah is part of, and with Uniprot that Rosanna is part of, and many others. So for me, it's really exciting because I work across so many different domains of molecular biology, bringing that data together in the work that I'm doing. And it's also quite interesting and challenging because we work so closely with the developers to build and maintain the databases, whole technology behind it. And you really have an influence in how that works, because at the end of the day, the curators are also users of the databases and the tools that we create, and we're all users of each other's databases. So I like that whole aspect, and working with people as well, not just data, working with people as well.
And so another question is from Deandra. Sorry if I pronounced that wrong. Was how do you design efficient workflows to delegate a list of gene entities to multiple annotators for manual curation? So I think this is something you can speak to, Rosanna, how the curators choose their work or triage their work.
So as I mentioned during the talk, we have some priorities, like we do a lot of curation of human protein entry and model organisms, and we have also the user requests that are a priority. We handle them first, and then it's also at least in my case, is also my expertise. I curate enzyme, and also if I found, I don't know, a protein family that I'm really interested in. For example, now I'm curating a lot of enzyme in the parasite for malaria, because I just, yeah, it's just the interest in the field that make you choose the entries. So it depends. We have some guidelines, but I have to say, at least in Uniprot, we have some freedom to choose what we want to curate.
Yeah, and so there are technical solutions within Uniprot as well to make sure that curators are not overlapping each other on resources, and also shared with other people such as Birgit to know that they are also working on the same entry and let each other know. And there are technical solutions from that. So Sami asked, she says, I want to be a biocurator and give a good contribution from my side. What would be a suggestion for me as a beginner? Well, the first thing I think you should do is go to the biocurators.org website. There is a biocuration society, and there is an awful lot of help there and available jobs and the training that you should take to become a biocurator. So please start from there, but do reach out to any of us after the call as well. Charlotte has made a very good point of, should we impose standards in publications? And I'm going to give that to Birgit.
Thank you. Well, the short answer is yes. The long answer is other people at EBI, for example, our literature services, have spent a lot of time talking to journals, and it's just not very easy in practice. And it's small steps like sequences are now mandatory to be submitted, for example. Structures, I believe, have to be submitted. Many journals have got those as prerequisites for accepting a manuscript. And then there's some domain-specific journals that also require you to submit your data. So, for example, apologies. There's some proteomics journals that won't take your paper unless you've submitted your data either to Proteomics Change or IMEX Consortium, whichever one is applicable to you. So that's down to the journal that actually serves a specific community. And also the reviewers can help. So if you review a paper and if it's a single Western blot, it's probably not worth submitting it unless it's something incredibly new. But if you have large scale data or a reasonably large scale amount or something that's very new, then it's always good to go directly to the databases and ask if you can submit it, and they can tell you if it's not for yours, maybe it's for another one. And also if you act as a reviewer of a paper, you can reply to the authors or directly to the editor and say this bit of data should really be submitted as part of the acceptance of this publication. I don't know if I covered everything, Claire.
So this is a passion for all of us, is the answer there. And if there's anything you can do to help us with it, we'd be delighted. So another question was from Karen, who asked, what are the conditions? I'm not quite sure what the question refers to, but if it's how much we get paid and where we work and our environment, it varies very much across the resources. But again, if you go to biocurator.org, there's a lot of information there from surveys done across biocurators around the world and how they work. And that will help you in those decisions question. So then Rodrigo had a very specific question about annotating genomes. So my question to him would be, does he know about Ensemble, which is another EBI resource? There's a similar one in the States as well, where you can upload your transcriptomics data on top of the genome and help improve it. So then they would do the annotation for you and tell you what the genes are. So please have a look at the Ensemble resource, ENSEMBLE. And there's again tutorials about how you actually do that on the website. So Dinanda again asked about how you handle protein genes street names while literature mining or data handling. Oh, the street names are one thing. You don't go anywhere near flies if you want to hang on to your sanity for data mining. There is now far more stricter rules on the nomenclature of genes across most organisms to make sure that this doesn't happen so much. So another one for you, Rosanna. Marcella says, I interpret large sets of genes and love using both UniProt and genecards.org. Do you know how both databases compare regarding the frequency of updates, etc.?
So to be honest, for GeneCard, I don't know. For UniProt, the data is updated normally every 8 weeks. We are a bit behind for this current release, but usually every 8 weeks it is updated with new entries. All the cross-reference also to GeneCards are updated. But for GeneCards, I don't know, unfortunately.
It should be in their help documentation. Most people write it in there. Like was another question I answered about how do you know how to submit? All resources have lovely help guides that most submitters never read. It's heartbreaking for the curators because it's all documented there under help. I sent the link from my resource, just if you feel the need. Does text mining help currently play a role in the process of data curation? It does in automatic, that's from Alan Barron. It does in automatic annotation, and UniProt uses this to a certain degree. And then anyone else? Deborah, Brigitte, do you use data mining?
So we don't, because most of our data is in figures and tables for molecular interactions. So it's really resource specific. There are many initiatives that are trying to use various versions of natural language processing to pull these things out by using keywords that describe interactions or methods that capture interactions and things like that. There's a lot of work going on, but nothing yet that can be just straightforward implemented into our workflow. I think, Rosanna, UniProt have something that you use for triage, right?
We do. We use PubData that does a bit of text mining on the PubMed publication that helped to identify publication, but it's still we still rely on manual finding. But I think at least a while ago, the worm-based database was doing text mining for of literature to come up with their function of the genes that they have. I don't know if they are still doing it.
There's one last one. How are older publications approached in curation? They're not so for a text suitable document or to be translated, but I know when I used to curate enzymes and structures, they were very much, the old papers were the best papers because they were the pure biochemistry, chemistry papers, and you will still see people adding those in, but that's manually done.
Okay. You have a new question from Colby?
Yeah, I was going to type also answer live to you, Rita, about pre-publication data. Yes, all databases love to get your pre-publication data, and we can keep that private until your publication is out. So just contact the relevant database, look at the, like Claire said, check how to submit it, or just hit the contact us button if you have any questions, you can't find the submission page or link, and then we'll walk you through it. No problem at all.
So, sorry, Alan, aside from pre-publication submission, are there ways to speed up biocuration? Depending on the database, really contacting them and saying this is a priority always helps curators triage things. So I would recommend you do that. I know our curators definitely work like that, and Uniprot. And as Deborah explained in PDB, it is a completely mutual beneficial relationship to build. Nicole had a question about street names. Quite often gene names were not really specific. They could be things from English or the language of the original name of the scientist, and so it's difficult to distinguish them from the language that has been spoken in the paper aside from the gene name identifier itself. That's why we really recommend that you use the identifier as well as the name, as Birgit showed you. That really helps us find them really quickly.
And Excel sheets change certain gene names to dates.
Yes,
so
Excel is evil.
Yeah, so that's another thing to really be careful with. We take Excel sheets as part of our submission system, but we're aware of that. And if you can already map it to standard IDs, that helps.
There's just one new question about, are there any efforts to make biocurated resources multilingual?
Yes, so my one, for instance, is available in Chinese because we had a Chinese member of staff who was very helpful and did a lot of mapping work. So at least the publication guides, the submission guides were also in Chinese, but the underlying data frequently is in the language of the home country, and quite a few of them use them in English.
So, right, thanks a lot. Thank you all speakers of the day, and thanks to all who joined the webinar today, and thank you for asking lots of questions. And if you have a question in future, if you can think of any other question, you can write directly to our speakers using their email ID, which we have it in the presentation slides.
Hi, thank you.